Is it Mac?
Yes.
Oh, excellent. That was good, because I was getting worried I'd have to give your talk for you. And I'm sure it would be better if you gave it. So, Mac, thanks very much.
No problem. All right, the other presentation in the other room was running just a bit late, so there'll probably be some other people that are coming in while I'm talking. So today I'm here to talk to you about antibiotic resistance in Staphylococcus aureus that come from livestock, especially pigs. So let me begin. So Staph aureus, it's the leading cause of skin and soft tissue infections, pneumonia, and bacteremia in a lot of the developed world. In North America alone, we see it causing more than 500,000 infections that require hospitalization each year. So it is a major cause of infection. It causes a wide range of opportunistic infections, and it is something that a lot of people have become concerned about, especially considering it is acquiring resistance to a lot of antibiotics. So the strains that are resistant to antibiotics are concerning because we have treatment failure due to resistance, which may or may not increase risk of mortality. We have increased hospitalization, which adds to healthcare costs because people are staying in the hospital longer and require more complex drugs that might be more expensive. And just that whole complex therapy in general is more expensive. So one of the particular strains that a lot of people talk about is methicillin-resistant Staphylococcus aureus, or MRSA. This is just a nice diagram from EARSnet in Europe. As you can see, there's some regions in Europe where the proportion of clinical cases caused by Staph aureus, more than 50% are considered methicillin resistant. So it's kind of concerning, and especially for nosocomial diseases in general acquiring resistance. And here is a figure of deaths caused by different infectious diseases and non-infectious diseases in the United States in 2005. You can see compared to MRSA, which is right here. You can see HIV, viral hepatitis, influenza, tuberculosis, salmonella, just the relative burden it has on our healthcare system, on causing disease in our communities in general. It is relatively large. So livestock-associated MRSA. Some livestock act as reservoirs for MRSA. The big one, or one of the biggest ones, is pigs. So 40 to 50% of Ontario herds are carriers of MRSA, and pig handlers, about 25% are carriers as well. Whether this is long-term carriage or just temporary, it's still kind of debated in the literature, but if we compare it to the general population, at one particular time we only see about 1% of the general population testing positive for MRSA. So we have a lot of MRSA in the pork producers and the pig handlers and the pigs themselves in Ontario, and this is seen globally as well. However, pigs are rarely infected. We do not see MRSA having a significant burden on the health of pigs or swine production in general. It does cause mastitis in cattle. And in the media, we see these depictions of pigs or other livestock and talking about superbugs and antibiotics and making bacteria resistant and that's its implications to public health. So whether it does pose a risk to public health, there is some risk. I will say this: there is some risk that it does pose to public health. The magnitude of that risk is still unclear. There's people where there's been epidemiology and molecular testing that has confirmed people have died from livestock-associated MRSA, and there's been infections as well. So it is there. The magnitude, though, is still unclear. Antibiotics in livestock. So this is where this comes into play. There's the mantra of conserving antibiotic activity for human medicine, pressure to reduce antibiotic use in food animals, agriculture in general. And the CDC has different diagrams and stuff. This is just one of them about the spread of antibiotic resistance within populations, and you can see here that animals are implicated as a significant source. Whether the magnitude of that source of animals relative to the hospital here may be kind of skewed, but still, there's this pressure to reduce antibiotic use in food animals. So for pigs, there's been this lots of development in antibiotic alternatives, and this is just a chart of some of feed additives and different management practices that are considered antibiotic alternatives, and their efficiency at replacing antibiotics. And one of the top ones is zinc oxide. So in pig farming, zinc is used quite a bit. Pigs required it at 50 to 125 parts per million in feed for normal growth and development. But it is also used therapeutically at 2,000 to 3,000 parts per million in feed for controlling colibacillosis, so post-weaning diarrhea caused by E. coli. This is usually used in a 2 to 3 week period just after weaning. We refer to this as zinc therapy. Antibiotics for controlling the same disease are used at about 100 to 300 parts per million in feed for the same time period. So about 10 to 20 times more zinc is needed than antibiotics in order to control this condition. So how common is zinc therapy in Ontario? So a survey of 22 nursery herds from 10 counties, 73% of them use more than 2,000 parts per million of zinc. But there was also the risk of zinc toxicity. If you look here, this one producer was using it at 7,000 parts per million. This is from our study, but this is not the record. The record in Ontario so far is 9,000 parts per million of zinc. If you're using it at over 3,000 parts per million, it's going to be toxic to the pig. It's going to have toxic effects and cause hemorrhaging in the organs and the brain, and yeah, it'll kill the pigs. It's not good. So we really want to educate producers and nutritionists that this amount of zinc in the feed is not good. More does not mean it's going to improve growth and be better. So how much zinc is used in Canadian pig farming? Well, just an extrapolation based on these results suggests that there's over 300,000 kilograms per zinc each year that is used just therapeutically. That does not include the normal zinc used in the diet. And if you were to compare this to antibiotic usage in the USA, not very good numbers in Canada, but if you look at these numbers, you can just see how much zinc that is relative to the amount of antibiotics that are used. So what's the problem? So in about 2009, 2010, it was kind of discovered that MRSA from pigs are very common carriers of a zinc resistance gene, which is genetically linked to other antibiotic resistance genes within a mobile genetic element, as you can see here on the screen. I just kind of showed one here for simplicity's sake, but there can be multiple antibiotic resistance genes within that mobile genetic element. And then it's located within the chromosomal DNA of MRSA. So if you were to think about it, and we have a population, it's exposed to a selective pressure, such as high levels of zinc. It'll kill the ones that aren't able to survive, or almost all of them, and it will leave the ones that are able to survive, which carry the resistance gene. They'll be able to propagate and establish a new population. And the theory for this this kind of selection is the co-selection mechanism, where we can expose a bacteria that is resistant to one compound, and it causes propagation of other resistance genes. So there's a hypothesis that zinc may co-select for antibiotic resistance genes even in the absence of antibiotics through this co-selection mechanism. We did 2 studies to try to analyze what effect zinc may have in this whole antibiotic resistance pathway and the propagation of antibiotic resistance within MRSA and pig farms. So the first was a randomized controlled trial. We took a herd of 110 naturally colonized pigs that had MRSA, split them into 2 groups at weaning, and gave them 2 different feeds. One feed was a control feed, just having 100 parts per million of zinc oxide in it, and the other was a treatment feed with 3,000 parts per million. Both rations were antibiotic free. And then after 4 weeks of sampling, we looked at how many pigs in either group were carriers of MRSA during that time period. So just a summary of the results from study number 1. The randomized control trials showed that throughout the suckling period, there was a pretty stable prevalence of MRSA at about 5%, and there was rapid turnover. So basically every sampling, it would be new pigs that were positive. And at weaning, there was a major spike in the treatment group, and then there was a delayed and minor spike in the control group. And the statistics show that it was significantly higher for the first 2 weeks after weaning in the proportion of MRSA. And then after that, it kind of declined to the point in the control group where it was undetectable, but it was still detectable at about 5% in the treatment group. And we typically see it persisting at that low prevalence until slaughter. Looking at the persistence of MRSA within each group, so in the control group, you can see that so the negative is they never tested positive throughout the entire study. Transient is they only tested positive once, and persistent is they tested positive multiple times, so 2 or more times. Within the control group, 74% of the pigs did not test positive at all, whereas in the treatment group, 34% were negative throughout the entire study. But then the persistence is where we see a big difference. 2% carried it multiple times throughout the study period in the control group, whereas 22% in the treatment group were persistent carriers. Study number 2, this was a prospective cohort study. So we went to 26 different farms, had a total of 390 pigs that we sampled. We looked at them just before weaning, and then 3 weeks later, we did a survey for potential risk factors, collecting a bunch of farm information. We collected drug and feed records. And here are some of the results. So the blue are the MRSA negative farms, and the red are the positive farms. Prevalence was about 40%. And you can see that it's kind of everywhere. It's not really localized to any one area. So the summary of these results, so 90% of the MRSA were resistant to zinc, and farms with MRSA were more likely to use zinc therapy. On the left-hand side, you can just see the simple univariable analysis, and then multivariable logistic regression model predicted that pigs fed 3,000 parts per million zinc are 12.4 times more likely to carry MRSA than a pig fed 250 parts per million of zinc, and it was very significant. And then surprisingly, we did have this as a risk factor, and we did look at it in our survey and everything. But MRSA was also associated with frequent disinfection of nursery pens, and it was slightly associated. There was a tendency for it to be associated with frequent disinfection of the corridors as well. Many disinfectant resistance genes were found within these isolates, particularly for quaternary ammonium compounds, QACs. So all isolates carried at least one gene. 75% carried 2 or more genes. And we saw fairly high levels of resistance when we tested them as well, and a bunch of genotypes, with the second last one being the most common at about 35%. And in the literature, it's been reported that this can occur through the same post-selection mechanism as well. Similar findings have been reported for Salmonella and E. coli, so strong association between heavy metal tolerance and antimicrobial resistance in Salmonella serotypes important to public health, and zinc feed supplementation of piglets increases the proportion of multi-resistant E. coli in vivo. So the take-home message from this is that zinc therapy, although marketed and used as an antibiotic alternative at very high levels, may be contributing to antibiotic resistance. So kind of counterproductive. But there's also other factors that can be contributing to antibiotic resistance as well. not just antibiotic use, but heavy metals, disinfectants, the microbiome, behavior, environment and ecosystem, social determinants, and some unknowns. These all play a critical role in the antibiotic resistance crisis, and we should consider them all when we are doing our studies and making policy. Just an acknowledgement, thanks to Dr. Friendship, Dr. Weiss, the people who helped out in the field and the lab, and the funding, and particularly all the producers for their participation. All right, thank you.
Thanks very much, Mac. Any questions for Mac? Yeah, Doug.
Yeah, so if 25% of pig handlers sometimes can have MRSA, do you know which direction it's moving, or is it in both directions? Can you repeat the question?
So he asked if 25% of pig handlers have MRSA, do we know which direction is it moving in? So is it moving from human to pig, or from pig to human, or both? So a lot of the MRSA we get from the pig handlers is our livestock-associated strains. So there's tests that kind of differentiate which ones are associated with the livestock and which ones are associated with the humans based on their adaptability to either species. So a lot of them are the livestock-associated, but there's been a lot of research that suggests that they're not actually able to colonize the pig farmers long term. It's not colonization, it's more of a contamination issue. So because it's become more adapted to pigs, people who go into the pig barns, they just kind of become contaminated through the environment. They'll carry it for 24 hours and then they'll be negative. So.
If pigs are being treated with 3,000 or higher doses, does that have any impact on concentrations in meat? And are there any food guidelines in terms of acceptable concentrations of zinc in things like pork?
So he asked if pigs are being treated with 3,000 parts per million of zinc, does that have any impact on meat and zinc residuals in the meat? This therapy is used during the nursery, so from weaning till 3 weeks after weaning. So then it's a long time till market. And they're only able to absorb so much, and a lot of it is excreted into the manure, and then it gets put into the environment. So that's also something to consider.
Any other questions? I had a slightly provocative question, and that is, if you were giving the same talk to pig producers, and a pig producer said, "So does that mean we should stop using zinc on pig farms?" What would your response be?
They're coming up with better products. So being able to use zinc at lesser concentrations, but it's been specially formulated so that it bypasses it doesn't start working till it gets into the intestines. So you're able to use a smaller concentration, and there's been studies that have shown that there's a dose response between zinc and MRSA. So I would kind of advocate going for those better products that still work at lower concentrations. Yeah.
Any other questions? Maureen?
The pig producers will say, aren't those more expensive?
It's not, zinc in general.
10% is cheap.
Yeah, zinc is even cheaper, but
Enteric-coated zinc is going to be more expensive, so they're not going to want to use it.
I don't know about the price of it right now, but if they're legislating bans in Europe for zinc and stuff like that, then they're not going to have a choice.
Any other questions? Okay, Mac, thanks very much. Can you please join me in thanking Mac and the other two speakers for their talks? Thanks.